So it's all about CRISPR. I'll talk about CRISPR all day long. I could talk about CRISPR for 2 days on end and maybe more. And CRISPR is one of those technologies that has really allowed the genome editing revolution to become a reality under a timeline that's really impressive. And I'll talk about CRISPR as a technology useful for ag and beyond. I'll talk about many different applications of CRISPR and CRISPR-Cas systems for food and for ag, and most importantly, about opportunities moving forward. What I'm going to try to do here is in 15 minutes, and I'm going to give a different talk, because some people have heard me talk before, so I got to keep people on their toes all the time. What I'm trying to do is in 15 minutes, kind of try to convince you that it's a great topic. CRISPR-mediated genome editing, it's very hot, obviously. Now is a great time, and some of you may have heard already about the CRISPR craze, and the CRISPR craze has lasted for 2 years already. And every year, and every time I give a talk, people think it's over, and I think it's very much alive. I think here it's a very much attractive place for CRISPR to make a difference in ag. And we hear about the Boston-centric CRISPR world on the East Coast, the San Francisco-centric CRISPR world on the West Coast, but most of those people really focus on translational medicine. I think there's a huge opportunity for the RTP to really play a big role in ag and biotech. And obviously, we have a lot of players and stakeholders in the audience. I've met many of you. I've talked to some of your companies, and there's a lot of excitement for this technology and the field here and in the audience today. And I'll try to show you how there are tremendous opportunities for the business, actual deliverables that are relevant for trade enhancement, generating lines, models, doing R&D and applications. And I'm going to try to illustrate that many people have already studied that there's a great potential for high ROA and high ROI in the business world. And I spent 10 years in business working on the CRISPR at DuPont. And now at NC State, I have a little bit more freedom to operate and a little bit of a wider scope. So I'm not going to talk much about science per se, but I can't talk about CRISPR without telling you what CRISPR actually is. And there's many a CRISPR expert that aren't familiar enough quite with CRISPR to know what it is, where it comes from. And CRISPR is the adaptive immune system of bacteria. And there's only 3 things you need to know about CRISPR, is that it's DNA-encoded adaptive immune system, whereby the immune system picks up DNA from invasive elements like viruses, so DNA-encoded. It's RNA-mediated, and this is where the single guide technology comes from. The single guide RNA technology comes from. It's RNA-mediated, DNA-encoded, and DNA targeting, whereby the DNA-encoded RNA-mediated machinery drives nucleases towards sequence-specific double-stranded break genesis in any organism of interest. And this bacterial immune system has been co-opted into what's called the CRISPR or the CRISPR-Cas9 technology for genome engineering and genome editing. And essentially what CRISPR is a Cas9 nuclease. It's an endonuclease that allows you to act as a molecular scalpel and cut DNA very precisely in any genome and any cell of interest. And one of the reasons why it's so popular, and so convenient, is that it has a lot of advantages. It's programmable, it's specific, it's transferable, it's efficient, it's precise, it's affordable, it's quick, it's multiplexable because you can have multiple guides and multiple Cas9 systems. And it has been adopted at a really, really, really fast rate because of all those advantages. That being said, it's only a 2-year-old technology to some extent. And there's still some gaps and needs that the technology needs to address and the people need to address to kind of take that to the next stage and engage into the future from a commercial standpoint, so product developers can really harness that to the best of their abilities. And Cas9 is a little large. There's PAM-dependent targeting, motif-dependent targeting. There's some off-target cleavage concerns, though they've been heavily exaggerated, arguably. And then different guides have various efficiencies. And how you design your guide and how you select your guide is very critical into efficiency. And last but not least there's a wide range of applications already for which proof of concept have been made for ag, for biotech, and for medicine. So it's a very broad-ranging range of applications. I'll walk you through this. And then beyond genome editing per se, as rewriting the genome, you can actually do different things that I'll walk you through in just a minute. So if I look at genome editing 1.0, right, if many, not all of you are genome editing experts in the audience, I wish it were the case, but perhaps it is not. What genome editing is rewriting the genome. That's what you do. You edit the sequence content of your genome of interest, and what you do is you do so by generating what's called a double-stranded break. Okay, there's different molecules you can use to guide Cas9. Think of it as Cas9 as a scalpel, and the guide RNA as the driver of where that scalpel is going to cut. Okay, so you generate a double-stranded break, and for the cell to survive, once your DNA has been cleaved precisely, you need to repair it. And if you repair it to the wild-type sequence, it's going to cut again, and you're not going to make it. If you repair it to something else, you're going to survive. And that something else is based upon which kind of repair pathway, non-homologous end joining or homologous-directed repair or AEGA, macromolecular-based joining and whatnot. But various different repair mechanisms within the cell will allow you to reattach the DNA after the cut. And you can do so imprecisely. Using an HEGA is going to leave a little bit of a scar, but it's going to work. And you either will have a small mutation here or a small insertion or deletion at the site of cleavage, but you cannot quite control what the end result is going to be. It's kind of quick and dirty way to edit your genome. Or you have the elegant, more surgical way to edit your genome called HDR, homology-directed repair, whereby you give the cell a template and you tell it how to repair that gap, that double-stranded break in the genome. And you can induce or include here a design mutation that's very precise, or a design insertion, or a design deletion. Okay, that's the basis what Genome Editing 1.0 is. Now, if you fast forward to 2015, this was so Genome Editing circa 2014. If you fast forward to 2015 or the future, and Genome Editing 2.0, it's really not just changing the actual sequence of the DNA. It's not just changing that sentence when you read it. It's changing the punctuation, where you put the emphasis, and other things. So you can delete things, insert things, knock out things, but you can use a deactivated version of Cas9 to actually bind DNA and either induce transcription or repress transcription. Okay, there's a great series of papers that came out. There's a nice paper coming out this week out of Charlie Gersbach group at Duke, showing you can actually fuse Cas9 with effector molecules to rewrite the epigenetics of a genome in human cells. You can use Cas9 fusions to recruit things of interest to a location of your choice, or by engineering, or you can tether Cas9 to fluorophores that allow you to actually gaze into the physical imaging of sequences, chromosomes, histone, chromatin, new chromatin, heterochromatin, and others. That's really the genome editing revolution, circa 2.0. Now, if you fast forward into what actually has happened, and use the past to predict the future to some extent from a scientific standpoint, CRISPR is one of the hottest topics in science altogether. Not just biology, not just genetics, not just genomics, not just molecular biology and sciences and the like, but across all of science. If you look at the pace at which papers are getting published and or the pace at which the CRISPR literature is getting cited, it's just unbelievable. It is faster than RNAi, faster than PCR, faster than TALENs, faster than almost any topic in biology in the history of publications. And this is arguably unprecedented. It's a very disruptive technology. It's adopted really, really fast. And what's amazing is that, with the CRISPR craze of 2013, this is kind of where the tipping point occurred here. Okay, people are already winning millions of dollars in prizes and awards and fame. And it was Discovery of the Year in Nature. Recently, it was runner-up at Science. And almost every week, you get a paper in Science, or Cell, or Nature, or Molecular Cell, or NBT, and half of the time they make the cover. Okay? And in some cases, it's hard enough that within 6 months of publishing a paper, those publishers will republish the whole series dedicated to CRISPR-Cas systems. So this tells us that it's cool and interesting from a scientific standpoint, but beyond the science itself, there's a lot of interesting items that make it compelling. And one is just the general public. The press is running with CRISPR stories all the time, occasionally perhaps more so than some people would hope. And not all journalists do an equally good job at covering the right stories or giving the right details, but we'll have our agendas. But if you look at right now, things like the New York Times, NPR, Twitter, YouTube, Forbes, The Economist, Time Magazine, The Guardian in the UK last week, you can see CRISPR everywhere. It's not just the scientists that are jumping on the bandwagon. It's also the news. That being said, we have a lot of people in the audience who may not care much about the science, though they are scientists, who may not care much about the news, though they are consumers. You have to look at it from a business standpoint. And forget about the science accolades and forget about the cool stories. Let's look at the business, right? And if you look at CRISPR as a business, it is likewise arguably unprecedented in many ways. And we saw commercial products get on the market within 6 months of the first paper showing it could work in human cells. 6 months, right? We all know what product development timeline can or cannot be. in the industry, and you had some risk-averse large companies getting in the game at the outset. And between January 2013 and the summer of 2013, very large companies like Sigma, like Life, like others, had products on the market. In an environment where IP is questionable, in an environment that's very competitive, and the reason those people decided to get in, and many people thereafter decided to join, is because it is compelling. And if you're not in the game, you are not competitive. And this applies not just to biotech, but also to pharma, also to ag, also to food. And what's really amazing is not just the business trifecta across different segments, but also the kind of companies that try to get in the game. And we're talking about Fortune 50 companies, we're talking about mid-sized public companies, we're talking about startups, we're talking about venture capitalists getting in the game, and we're even talking about not-for-profit organizations like AdG. Okay? And only rarely and really seldom do you see interest and all those different kinds of companies getting into the same game at the same time with the same objectives. If you're not interested in the business, and you're not interested in the science, and you're not interested in the news, you can look at what some of the arguably smartest people on earth are deciding to do with their money. And look at what the VCs are doing. Right? Talking about some very, very smart, very future-looking people. I'm not going to name any names, but we all know who they are. And those people are deciding to put tens of millions of dollars into a technology. Though it's competitive, though the IP is challenging, though it comes with many strings attached. And you can see Editas is a great example, $43 million, CRISPR Therapeutics, Intellia Therapeutics, Caribou Biosciences made the announcement last week. And we start to see some very large pharma companies make public announcements with regards to their eagerness to get into the CRISPR business. So, the science and or the news and or the business and or the investments really indicate that this is a hot topic right now and people ought to get it. So why is it and where's the technology headed? So one of the things that you need to know is you only need two things for CRISPR to work. One is the Cas9, the nuclease, and one is the guide RNA that drives it towards sequence-specific targeting of complementary sequences and genesis of a double-stranded break, precisely 3 base pairs away from PAM. And it's only a 2-year-old baby again. So you need to optimize your guides better, how you design your guide, how you have guide composition of different modules, and or how you use different in silico tools to select or pick your guide still needs much improvement. Okay, it works well, but it doesn't work 100% of the time. And people ought to know a little bit more about why it works when it works well. Likewise, in addition to the guides, much work needs to be done on the nuclease called Cas9. There's a lot of people working on next-generation Cas9s that are skinnier, that are smaller, that are more efficient, that are meaner, that are more packageable, and the like, and also different Cas9s that can work together. So if you're interested in doing more than just one thing at the same time in the same cell, you can do upregulation and or downregulation and or epigenetics and or editing and or nicking and or imaging, you need orthogonal Cas9s that don't crosstalk. Okay? So we're actively working on diverse sets of Cas9s. You need new PAMs to do different GC content targeting, more technical here. And also if you think of ag, right, you need to start having the ability to harness more palatable, more friendly Cas9 derived from more copacetic organisms. And right now, one of the challenges for the first generation of Cas9 is that they are derived from non-attractive human pathogens, and some people are working on systems derived from generally recognized as safe bacteria that are used widely in the food supply chain. And in addition to the guide RNAs and nucleases, there are still some outstanding issues that need to be improved or further enhanced, like delivery methods, safety, toxicity, and regulatory assessment of Cas9s. There's a lot of people trying to work on GMO, non-GMO alternatives of Cas9s, how you deliver it, where they come from, how you harness them, and can you do Cas9 without doing DNA? So we're going to talk about that. Come talk to me after the talks here. And then last but not least is IP. There's a lot of people that know that IP is challenging, but there's enough potential here that it is a competitive situation. So if you look at the next generation Cas9s, what we're trying to do here is really try to look at different orthogonal sets of Cas9s. And one thing we're working on here just down the street at NC State is very much this, is looking at Cas9s that are derived from lactobacilli, beta-cocci, enococci, some non-pathogenic streptococci, and bifidobacteria that are commonly used in the food supply chain. And what those things are, they're very diverse in their sequences. This is just a phylogenetic tree of Cas9. And then some of those are much smaller than the current incumbent Cas9, which is a little bit hard to package. As we characterize those different Cas9s, we characterize the guides that direct them and that really allows them to selectively target DNA in your cell of interest. And then also we work on the targeting space. And for those of you who are familiar with the PAM, we try to target different PAMs with different length, with different GC content, and with different targeting spaces. So regardless of your GC content and regardless of the sequence that you're trying to target, you can find the Cas9 that goes with it. Now, one thing for ag that is yet to really hit mainstream is the part of crop traits and health that is linked to the microbes that are in the environment of the plant. This is a very hot topic. There's a lot of high-profile companies investing in microbial applications, and CRISPR so happens to be a microbial system used in bacteria, from bacteria, for bacteria, that has a lot of potential for a number of eukaryotes and beyond. But if you look at applications in bacteria, you can use the same two-component system or the native system to type the strain, understand where it comes from and how they relate to one another, to enhance the resistance or to vaccinate a strain against the incidence of viruses. You can control plasmid DNA uptake, dissemination, or even eliminate plasmids from strains of interest that you may want to genetically manipulate. Or you can use Cas9 to target the chromosome DNA in different ways to do different things. Obviously, genome editing, transitional control. We are actively working on genome remodeling and really like taking out big chunks of genomes that you don't need or you don't want, and then start using the genome as a puzzle for more synthetic biology type applications for workhorse in the industry. And then last but not least is you can co-opt Cas9s to induce killing and suicide in cells, and they have tremendous potential as next generation antimicrobials. If we can have the next slide. So if you look forward, right, this is part of what we're trying to do here, look at technology of the future, there are still needs and gaps that need to be addressed by a lot of CRISPR scientists. There's thousands of them here around. So one is IP, really understanding the landscape, how to get to freedom to operate, and how to predict an unpredictable challenge. Increasing guide efficiency, increasing Cas9 specificity, understanding more how different parts of DNA repair can be triggered, so you can try to rebalance that towards homology-directed repair in a more surgical way, work on new systems, look at safety, look at toxicity, look at regulatory pathways, preferably towards non-GMO, obviously for ag and food, looking at delivery methods to enhance the potential of that technology. Biolistics, a huge potential for plants, obviously, and RNPs is the way of the future for DNA-independent genome editing. Cas9 can be fused to many different proteins to do different things, like epigenetics is the news of the week. You can combine a lot of the knowledge that we have from RNAi with the CRISPR-Cas9-based applications to really enhance guide synthesis, guide design, and guide applications. And then last but not least is, there's a lot more to CRISPR than just Cas9. There's a lot of those Cas proteins that hold tremendous promise moving forward for genome editing and beyond. So with the last slide I have, what I'm hoping that I have done in 15 minutes is to really convince you that CRISPR is a great topic. Obviously, it's a timely topic. This is the right place to do ag. I think North Carolina has a tremendous opportunity to be in the leadership position for sure. There's many players in the room here and close to the RTP or within the RTP that are actually very much leaders in the field, needless to say. Great business opportunities, which is why people are investing in this technology, great deliverables for trait enhancement, and then obviously a very sound investment. So I hope you're going to bring the CRISPR element to your lab or your project, and we'll take questions after the last talk. Thank you.